Okay, guys, welcome back. Season 1, Episode 4. This one's a really special one. I'm excited for today's podcast. I'm bringing to you actually an interview of one of my colleagues, who specializes in a field that I don't know too much about. So I thought it would be really appropriate to, I've never interviewed anybody before, so just interview this person. Let her teach me and all of us about today's topic. Today we'll be talking about gene editing. What that is, why do we care about gene editing, particularly with the angle of treating disease, mostly is how I focus the interview. And then, I think it's a really insightful interview, so I'm excited. So she doesn't play World of Warcraft, obviously, so I had to record the interview separately, but I also want to incorporate the World of Warcraft part. So I'm just going to be healing a key here, and then we're just going to listen to the recording. I may or may not pause it at certain places just to add a couple insights or like additional thoughts. But for the most part, I might just let it play and then add on at the end some additional points, just hit on some points that I took away from the interview, because I learned a lot. I thought it was great. I thought it went a lot better than I was even expecting. It was really, really good. She's very smart, obviously knows a lot about the field, and it was just great to talk with an expert in gene editing. So I learned a lot. Hopefully you guys learn a lot too. I'm just going to play the file here, so we'll listen to it. I'm going to be playing World of Warcraft as a Discipline Priest in a Mythic Plus dungeon. And there's a YouTube component to this as well, so you can watch the entire VOD on YouTube if you want. But we're going to listen to the start of this. I'll just be kind of quiet here, and then I'll add in some commentary as necessary. How about we just leave it at that? So I'm just going to start it here. So here's the interview from start to finish.
Okay. Morg. So maybe we shouldn't give away too many identifiers. I have no idea where this podcast is going. Probably nowhere. But just in case, I'll just call you Morgan. But tell me a little bit about yourself. Like, what's your expertise, would you say?
Expertise. I have a Bachelor of Science in Biochemistry. And then I've worked for 5 years at a gene editing company. So one of the first gene editing CRISPR Cas9 companies.
Sweet.
I'm going to pause it right there for just a second. I'm not going to do this very much. I think she was being too humble. She sold herself a little short. So on top of all of that, 5 years industry experience with gene editing, she's also into her 3rd year of her PhD, where she does similar work. And she was just recently published in Nature, the journal Nature, which is, the most prestigious scientific journal there is. So, yeah, she knows her stuff. She knows what she's talking about. I'm just going to add that in there as another bona fide. Okay, we'll continue.
So I have quite a bit of experience in the gene editing field, and specifically in the biophysics of how that all works.
Perfect. All right. Well, I'm excited to have you on because this episode is actually about gene editing, and that's why obviously I chose you. So again, we're just trying to keep it very simple, maybe, I don't know, 8th, 9th grade level type stuff. If I feel like it's a little confusing, I'll just stop you and try to figure out exactly what you're trying to say. But basically, what I see is we want to get a good definition of gene editing, and then what that is, why we care, and then move into CRISPR, that technology. Just briefly talk about that, what that is, because I feel like my type of podcast, I just want to get important information, scientific information out to the public, to where I feel like people should know what CRISPR is. I feel like it's that game changing. So let's help people understand what gene editing is, with CRISPR kind of the back end as the goal, the target to talk about. So gene editing, what's a gene, and why do we want to edit it?
So our cells are all made up with DNA. So every cell has DNA in it, and that's our genetic code. And this is what actually helps us function and do everything. So like your hair color, your eye color, those are all made up of different genes. So there's like 6 genes that do hair color or something like that. And so everything in your body, though, every function is related back to these different genes that is in this DNA. And sometimes there's things that go wrong in that gene sequence, causing problems down the line. So you've probably heard of different genetic disorders. One of the big ones is sickle cell disease. So people don't make the right type of red blood cells. And so it's really hard for them to survive because they can't hold on to oxygen. Their red blood cells just don't work the way they're supposed to. And so this happens at the DNA level. There's something wrong with the DNA. And so then there's something wrong with the genes that are produced from the DNA. And so then you get wrong red blood cells. And so you have different genetic disorders that are all linked to the DNA and these problems in the DNA. So the goal of gene editing is to actually fix these problems at the fundamental level. Because right now you can try and treat the symptoms and things like that, but fundamentally the body is going to make the wrong thing every single time. So if you can correct it to try and making the correct thing, that's what the goal is of gene editing, is taking the DNA that's wrong and replacing it with something that is right, so then you aren't making the right things.
Interesting. So if someone with sickle cell disease, just using that example, we can treat the symptoms of that disease, but that person for life is going to make incorrect red blood cells, and we can't fix that necessarily without gene editing. We can only kind of treat the symptoms of that. Interesting. Okay.
That's true. And typically treatments have to be done pretty regularly, so every other week or sometimes every month, and a lot of times it's shots, and I don't know about you, but I wouldn't want a shot every single month.
No.
That sounds awful. Or for a sickle cell patient, it's blood transfusions. And so once again, that's not a long-term fix, and that's an arduous process. It's not something a lot of people would like to do. A lot of people are scared of just giving blood, so I can't imagine having to go in and get a transfusion all the time.
True. Okay. Okay, so in medicine where we stand currently, how far, like how advanced are we in editing genes? Is it possible? Are we there yet? How far do we have to go?
It is possible. There are a few companies that are already starting clinical trials, so it's in the clinic. We've shown that it is safe in animals. We're showing right now that it's safe in humans. And so that's kind of the big step right now. That's where we're trying to get through what's called a phase 1 clinical trial. So is this safe in humans? And once it gets past there, then we know that proof of concept, this works and it's safe, and it can start being developed for all these different types of genetic disorders. So right now it sits in, we're trying to make sure it's safe. We think it's fairly safe for humans, otherwise it wouldn't have made it this far. But we're still waiting to see, are there long-term effects of editing a gene, since this should be a permanent fix. But you want to make sure for a long time, is there going to be anything else downstream that could cause problems? Is this going to be a good long-term fix like we want it to be? And so that's kind of where it's heading, is that we think it's safe for humans, we think we can use it for all these different genetic disorders, but now we really want to see, is it the long-term effect that we want? Because if it's still only going to be a short-term effect, like we're just treating the symptoms again, it's not really worth going through all the effort to continue this path.
So like a permanent cure is more what we're making sure that we're making it a permanent rather than just temporary.
Mm-hmm.
I see. Okay. In a general, very general approach or discussion, how do we go about editing a gene? Like, how exactly are we doing that?
So there's a few different ways that people have actually tried editing genes. The way we're going to mainly talk about is CRISPR oriented. And so you have this little piece of guide RNA that they call it. And so this guide RNA matches a sequence in your gene. And so
Okay, just to pause right there, we're just going to add on to what we talked about in the vaccine episode, right, where we talked about RNA as a therapeutic. This is the same idea, same concept of RNA. Now we're using it for gene editing. So remember in the vaccine episode, we talked about how RNA, we didn't understand how we could make it a therapeutic because it was so unstable, right? And so if we put it into the blood just as is, it would just get degraded instantly. Like, the body's really good at sniffing out foreign DNA or genetic material and just degrading it instantly. It thinks it's a foreign invader, right? And it just, no questions asked, just degrades it. And so for a long time, we knew about RNA, we just didn't know how to harness it as a therapeutic. And so kind of the first breakthrough idea was, okay, let's package this stuff up, this cargo, we call it, right, into some kind of delivery vehicle. And so for vaccines, you know, we use liposomes. And then she'll talk about a little bit more later in a second. But just to bring back some of the stuff we've already talked about. So RNA, for what she's talking about, same RNA, same ideas, unstable molecule, but it's what's used for gene editing, one of the tools used. So just to give you a little hindsight there. So I'm going to rewind just a titch just so we can get that question back.
Piece of guide RNA that they call it. And so this guide RNA matches a sequence in your gene. And so it's saying, hey, when you find this exact thing, this is where I want you to do your editing. So it's kind of like a barcode that tells you exactly what's there. This guide RNA does the same thing. It's looking for the same exact barcode to say, this is my match, this is exactly what I'm looking for. And once it finds that in the DNA, then the rest of the CRISPR machinery can come in and actually cut in the correct spots to make the changes that you want to see.
Interesting. Okay, so in a sense, we're looking for a specific gene by this barcode, as you call it. So when our system finds that barcode, we can start to do some manipulating of that gene. What are we doing to the gene? What exactly are we doing to the gene?
So it does depend on what you're looking at. So different genetic diseases and mutations are going to cause different things. So some manipulations are very simple. So sometimes you just have one tiny piece that's missing. So like one base of your DNA or one little piece doesn't fit quite right. And so you can just cut out that tiny little piece and correct it that way. Other things sometimes are much larger, and so it's much more difficult. You have to take out the whole gene and replace it with another one. And so that's when it becomes a little bit more complicated, where you have to engineer your guide RNA a little bit better, or sometimes you even have two, so then you can cut big chunks of this piece of DNA out, and then you're going to also introduce the correct chunk in the same spot. And so people have gotten really good at doing this in like bacteria cells, and we've been able to do that without CRISPR for quite a while, but it's really difficult to edit into a human cell. And so that's kind of where this challenge and where this technology has kind of advanced the field, is that it makes it a whole lot easier to do in human cells, which are much more complex than a bacteria cell.
Okay, interesting. Okay, so we brought up this CRISPR idea a couple times. Why is it so revolutionary? What's it bringing to the table? What's it making possible that wasn't possible before?
I think the biggest thing is that it's a potential cure for genetic diseases, but it doesn't have to just stop there. And so lots of people have tried treating symptoms of genetic diseases, but that's as far as you get. This would be a potential long-term cure. And that's probably the biggest thing that this technology has to offer, is that it has the potential to be a long-term cure. It's also a lot more simplistic than a lot of other routes that people have even tried to treat the symptoms. So there's a lot of different things that you can do to treat symptoms of genetic disorders, but typically they're just very difficult to deliver, get into the correct cells, get into the right area, and have long-term effects. This is much more simplistic. It's fairly easy to deliver CRISPR to the right types of cells, and it's also pretty easy to do the editing. The machinery kind of takes over once it's inside the cells. And so there's not a lot of work done on our behalves to get it in there and through, and then the potential of it being a cure instead of having to go in every month, every 2 weeks for treatment is going to be huge for that patient population. Not only for genetic disorders, though, too, you can look at most types of cancers. Cancer is typically a mutation in your DNA. It starts there. And so if you look at it from that standpoint, too, we can also start applying this to different cancers, correcting the cells that are doing what they're not supposed to because the DNA is damaged and is causing these problems. And so there is potential there as well.
Interesting. Okay. And then following up with that, so we go in and we can fix these mutated genes or these genes that aren't functioning correctly with CRISPR. Are there any potential risks, or are there foreseeable challenges or risks associated with this?
So there are always risks when you're looking at DNA and manipulating something so fundamental. And so since these changes are permanent, that is like a huge concern for everyone in this field of research. A lot of extensive screening is done typically before it even hits clinical trial stages to see if there's anything that could cause problems. At the company where I previously worked, we had a lead candidate for a while that we thought was going to be really good, and then we found out later downstream that it actually had a very toxic effect. And
what was that toxic effect?
Oh, it just killed the cells. They died.
Okay. So see, like something like that would make I think a lot of people kind of nervous, maybe a little timid. So how do we harness, or how do we like get the proper barcode or get the proper targeting? How do we dial that in?
I think it is a lot of screening and a lot of testing, making sure that one, your editing works well and so that you're getting the desired effects, but then also like long term, can you see the cells or the organism surviving, that there are no other complications? And typically people have also been adding in full genetic screens again. So at different time points, if you're editing a mouse and you want to see this one specific thing, but you're not just going to look for that one specific thing. You're going to look for all the different major genetic functions at the same time. And so you start screening for more things and broader things just to make sure you're not changing anything downstream and that it is going to be a good cure instead without having all these side effects. So there's a lot of screening that goes into making sure that no bad side effects are going to happen.
Okay, so I just wanted to just add a titch there. What she's talking about is actually super interesting. So we have the capacity to very easily sequence or identify the code of people's DNA, genome sequencing. If you remember like early 2000s where we sequenced the entire human genome, we can do that super fast now within probably 24 hours or even less maybe. We can sequence someone's entire genome. And so what she's talking about is if we are editing genes and we're changing someone's DNA, we want to make sure that we're changing what we think we're changing. So we have the capacity nowadays to just specifically, run an entire sequence for that patient, and that'll tell us, if we've edited off target, if we've edited somewhere where we didn't mean to edit. That's what she's talking about, screening, where they can get entire sequences of DNA and figure out exactly where it is that we edited the gene. Just a little background.
The other nice thing though about CRISPR is you don't have to reach really high editing efficiency to get a desired effect. So if there is
editing
potentially
efficiency, what does that mean?
So, sorry, I should explain. Editing efficiency means you have a population of either cells, or when you're looking at an organism, you want a certain percentage of it to actually get into the cells and edit correctly. So not everything is going to edit correctly. Not everything is going to edit, period. You could get the machinery into a cell and it can do nothing. And so that typically, for like a typical drug to work, you want like 90% of it typically to get into cells and work and do what it's supposed to. For CRISPR, depending on the genetic disorder, but specifically for sickle cell disease, you need about 5% to get into the cells and to work correctly. And that is all you need to actually produce the desired effect. So you can have a fairly low efficiency. So most of the time, if it gets into the cells, it'll either work or it won't. And you can have a very low efficiency of it working, and you'll still get the desired effect. Versus normal drugs, you have to have a good majority of that has to get in and work in order to produce the desired effect.
So what makes it so potent? Just only a few cells need to get affected, not all of them? Or is that how I should think about it, or a different way?
It is, I think, mainly because you are editing the genome, so you're editing the DNA. And so when you edit a few cells correctly, they're going to go on and produce more cells that already have that. So now their genome is corrected. So every cell that you're making after that, you're dividing and you're exponentially increasing how many are actually good and active and have the correct gene. So you only need a small amount because they're going to divide and make the correct things later as well.
Makes sense. Okay, okay. Okay, great. Yeah, this has been really good. Last kind of idea, I guess. Big picture. Where do you see this going? 10 years from now? 50 years from now? What's possible, do you think?
I think 10 years from now is going to be big on moving into some of these like smaller genetic diseases, and then also I think cancer is going to be the next big thing, because right now it's very localized. Most CRISPR companies are working on small orphan diseases, so things that are very rare that you can possibly cure. Sickle cell disease being the exception. A lot of people are working on sickle cell disease, so I think they're going to start branching out, and then moving into cancer is typically, it's a genetic condition. There's something wrong, mutated in your genetic DNA. And I think that'll probably be the next avenue. 50 years from now, it might be as simplified as I want my baby's eyes to be blue, so changing it to make sure your baby has blue eyes. Because people love cosmetic science.
Interesting.
And I think there's potential for it to be as commonplace and as easy to work with as that.
Wow. Very cool. Very cool to think about. Okay. Well, thank you. I appreciate you doing this, sitting down for this interview. I'd never interviewed anybody before, so yeah, I think it was really good.
Was okay.
Yeah, it was very enlightening. Very, very, very good. Thank you very much.
Okay, so that was the end of it. I thought it was insane. I thought it was so cool. She added so much like thought, and it made me rethink things, and just, oh man, it was such a good interview. She has so much insight into the field. Few things that I wanted to highlight there. This idea of potency. So I hadn't even thought about asking her questions about this. It just kind of came up. Where she's talking about how when you treat with like a chemotherapy drug in the body, you want as much drug as you can get to get to the cancer cells, you know? And she said 90%. I don't know what the numbers are. That sounds probably correct. Where you want like this high efficiency where all the drug that you put in, you want to get to the site of action. And that makes sense, right? Well, and then what's awesome and what she clarified on so neatly was for gene editing, you don't need that much efficiency. She said 5%. She said, you shoot for like 5%. And so I tried to clarify with the question. I was like, so what does that mean? Like 5% of the cells get edited, or like we have 5% of the genes in each cell get edited? And she said, you think about it like 5% of the cells get edited, and how that's enough is because then those cells, they're going to divide like cells do, and all of their daughter cells are now going to be edited and cured, like fixed, normal, healthy. They're going to be healthy now. And so all of the daughter cells into perpetuity are going to be healed, permanently healed. And that is why you don't need such a high efficiency. It's different, right? Like, so in cancer, in chemotherapy, you're trying to kill the cancer cells, whereas when you're doing gene editing, you're not trying to kill the cells. That's not the goal. You're trying to, edit or alter the cells in some way. You don't want them to die. That's not your goal. You're just trying to, make them better. Just fix a mistake. Which was a really interesting point, and that came up organically. I hadn't thought about that at all ever. That was a new concept to me, so that was really cool. And then the second thing that I found pretty interesting, well, a lot of it was interesting. The second thing that stood out to me was this idea of permanency, right? Like when we treat with chemotherapies or cancer in general, or any disease really. We're just treating a symptom, right? Like, we call it perhaps a cure, but we're just treating the symptoms a lot of the time and getting a patient comfortable. But in this case, or in gene editing, you can actually do like a permanent, lifelong change in their DNA code that makes it so they don't need treatment for their symptoms. Which I thought that was a really neat insight there, where she brought up the sickle cell anemia, where they have to go in for blood transfusions every week or every 2 weeks or whatever. Instead of that, now we're talking about a permanent fix, a cure. So I thought that was really insightful and just a cool thought in general, where we're actually to the point where we can cure some of these diseases that are giving people, real actual people, you know, a tough go in life. So that's just really inspiring. I love that idea. I'm glad she brought that up. Yeah, so I thought the whole thing in general was just such a good interview. I'm just really glad I interviewed her. She had so much insight. Yeah, I'm trying to think of. There was one other thing. I really liked this concept that the analogy of a barcode, right? Where a mutation happens somewhere in our genome, and our genome is humongous. I think it's what, 6 billion base pairs in a single person's genome. So it's just humongous. And any base pair can get mutated. And so to find that, to put something in the body that can locate the exact place where the mutation has occurred and fix it, is sort of mind-blowing, right? Like, you need some really good design. You need to troubleshoot. You need to be careful with, and you know, and companies do all of this. And I want to just, you know, kind of try to break the stigma a little bit of this fear of gene editing, because it's kind of like this, like a company is putting everything, like so much money into developing these systems. Yeah, it's profitable for them at the end of the day, but also they can't produce a crap product because it's all about the name, you know, like a Pfizer, for example, a big company like that, where if they mess up one time, they're done, right? Like their stocks plummet, they tank as a company. So they need everything perfect. They need it to go perfect. And that's kind of their long-term goal. Yeah, like, yeah, they're always thinking about money. There's probably like this money edge. But part of that strategy is to never make a mistake. And in order to not make a mistake, they need to do checks and more checks and rechecks and checks on their checks and make sure, absolutely sure, that there's no surprises, there's no adverse side effects, that this stuff is safe. And I don't mean to call out Pfizer. Every company is doing this because all it takes is one mistake to lose public trust, and that company is done forever. If you look at Johnson & Johnson, the vaccines, you know, there were 3 vaccines that got approved all about the same time, and then Johnson & Johnson had reports, I think it was like 7 reports of blood clots pulled instantly, like pulled, right? Can't find it in the States anymore. They had to start selling out of the States because no one wanted it anymore. That's just one, like, easy example, just recent example of where just any small mistake is going to cost you so much money. So they need to they have every incentive to make a good product that is safe, that does not have any toxicity. It sounds like I'm selling maybe for like a big pharmaceutical company or whatever, but no, that's the truth, right? Like, yeah, they're in it for money, and part of that money strategy is to not make a bad product. And so, yeah, there's a lot we need to learn about genetic engineering. We're just sort of on the precipice of something really big here, and there's going to be decades more of learning to do. But it was just really cool to get her insights about, 50 years from now, where do you see this? And she said, maybe we'll get to the point where it's so commonplace that you can choose your baby's eye color. And that's, we don't need to get into the politics of that, but just an interesting idea, right? Like, with decades more of research on top of what we already know, so much could be possible. Not only in terms of curing diseases like cancer, such a big disease like cancer, where we can actually go in and fundamentally fix the mutation. It just puts so much people out of misery and just helps so many people around the world. And then we can get to things as trivial as eye color. It's super cool, really cool concept, really interesting interview. I'm so glad she accepted to do it. And yeah, I thought it was a really great interview. So that's it for this podcast. Yeah, I loved it. I thought it was a really good one. So I'm going to try to get one out next week as well. But I like this type of interview style. It actually turned out really well. So I'm going to try to find other people, other colleagues I know who are willing to get on and just do a 20-minute interview and just pick their brain about some topic they're interested or they know a lot about and they can teach us about. So with that, thank you for watching. You can check out the YouTube component of this or the podcast component on Spotify or other podcast stations. But yeah, thank you very much for watching, and tune in next week. We'll be back with another episode. Thank you very much.